Hello, my name is Ladislav Vyklický. I'm head of the Laboratory of Cellular Neurophysiology at the Institute of Physiology, located in the beautiful city of Prague. I'm also a professor at the Czech University, where I teach physiology. I spent all my scientific career working on glutamate receptors, mainly using patch-clamp technique, and my colleagues will tell you about the projects we are involved in more detail.
In the brain, thinking and learning takes place at synapses, the functional connections between neurons. We use patch-clamp electrophysiology in precisely defined networks of cultured neurons. In the simplest case, a single neuron grown on a microisland forming synapses onto itself, or a pair of synaptically connected neurons. We study how synapses transmit different patterns of signals and how this process is altered when postsynaptic neurotransmitter receptors carry mutations found in patients with neuropsychiatric disorders.
My name is Bogdan Kiselov. I am a PhD student, and in my project I evaluate the functional characteristics of NMDA receptors with disease-associated mutations. For this purpose we use the patch clamp technique and recombinant NMDA receptors expressed in HEX cells. We evaluate such properties as agonist affinity, open probability, desensitization, and others.
My name is Prof. Vyklický. Here, our PhD student Mark Dobrovolsky and I employ an optical method called single molecular thread to study conformational changes of the NMDA receptors. We look how the NMDA receptors change their three-dimensional shape during the activation process. At the single molecule level, we can see how this sequence of conformational changes is different in receptors with mutations that are linked to various GRIN-related disorders. Our hope is that single molecular thread will help us understand structural and functional effects of GRIN mutations and therefore help us develop therapies against GRIN-related disorders.
We use zebrafish as a model organism to study the effects of neurosteroids and the consequences of NMDA receptor mutations. As a model, zebrafish has several advantages over mouse, which are rapid development, potential of high throughput screening, relative ease of genetic manipulations. We test the effect of neurosteroids using open field behavior test and pre-pulse inhibition. In addition, we have already created new NMDA receptor subunit knockout zebrafish strain with the use of CRISPR-Cas editing method.
My name is Miloslav Kořínek and I'm an electrophysiologist from Prague. My research is focused on endogenous sterols. These are compounds which are present in human brain and which modulate synaptic transmission.
I'm Agnieszka Kubik-Zahorodna. I work at Czech Center for Phylogenomics, where I lead neurobehavioral unit. Our team focuses on testing of transgenic animals that have mutated genes responsible for encoding NMDA receptor subunits.
Hello, my name is Aleš Balík, and together with my students, we are studying the genetics of NMDA receptor, and we are particularly interested how different variation in the genes regulate its expression, and also how the mutation within the NMDA receptor can change the trafficking and localization of receptor on the synapses.
My name is Jiří Černý and I'm doing structural bioinformatics and molecular modeling and molecular dynamic simulations of NMDA receptors, and I'm studying the effect of structural effect and functional effects of mutations in these receptors.